Oncology UK Cambridge Centre podcast. In this Integrated Cancer Medicine Research in Focus series, I talk to various ICM members about their research and how it is supported by the vision of the Mark Foundation Institute for Integrated Cancer Medicine. MFICM research uses cutting-edge analytics to maximise the use of diverse high-volume datasets and by capturing cancer heterogeneity in time and space in patients receiving active treatment. Integrated cancer medicine aims to transform the way the world treats cancer by affecting patients along their treatment pathway and ultimately accelerate cures. Today I have with me Jason Carroll, and we're going to talk about his research and specifically the work underpinning the Pioneer trial and also the outcomes of this clinical trial. Jason is Professor of Molecular Oncology and Senior Group Leader at the CRUK Cambridge Institute at the University of Cambridge. He is also co-lead for the Precision Breast Cancer Virtual Institute. So, morning, Jason.
Good morning.
To start with, please can you outline your role at the Institute and how you interact with the wider breast cancer community?
Yeah, so I'm a group leader at Cancer Research UK, Cambridge Institute. So one of the senior group leaders. I've been here right from the very beginning. So when the Institute opened in 2007, I was one of the junior faculty at that time, and I've been here ever since. What's that, 17 years? My lab's been almost entirely focused on breast cancer research, with the exception of the last few years. We can touch on that a little bit later. And a big part of that is not just doing research in our lab and learning about the fundamentals of breast cancer, but it's making sure, as you suggested, it's making sure that we make connections across the program. And I'd say at the beginning in 2007, the connections between the labs doing basic research in our institute and some of our clinical partners at the hospital were probably maybe not as strong as they could have been. But over the last decade or so, we really put a lot of effort and time into making sure that we can build bridges and collaborations. We do that on multiple levels. I work very closely with Jean Abraham, who is the clinical lead of the program. I'm the academic lead. We speak regularly. We have Joe Worley, who sits between us, mostly working with Jean, but we're chatting almost every week about various bits and pieces. We organize an annual breast cancer meeting, which we ran only a few weeks ago. And this is really to showcase the work that goes on in the breast cancer community in Cambridge, but also wider. We also have national and international speakers. And that's a really fantastic forum to bring students and trainees and postdocs and clinical fellows and faculty together. And we discuss everything from patient advocacy all the way through to fundamental research, clinical trials. And these meetings are a fantastic forum for everyone to get together. And what I realized is actually a lot of it is, it's not just about the academic and the intellectual connections, it's also about the social connections. In fact, it's really, really important. And there's one thing I've learned from my experience in science is sometimes the collaborations come from relationships that have been created through personal connections. And so the social events at the annual meeting is really important. We also do meetings across the campus. So I will present to Jean's team and I'll present to the breast cancer program. At their lab meeting, we will have some of the clinicians come over and join our lab meeting. Some of the clinicians have access to our building, so they come in as adjunct faculty. So there's free movement between the building, which is fantastic. I think one of the other really important aspects is this is something that we didn't do at the beginning. When I set up my lab, we were just a group of fundamental biologists, PhD students, postdocs, myself. And then over the years, we ended up, must have been probably about 12 years ago, we hosted a breast oncologist who came to do a PhD. And I just realized he just added a completely different dimension to the lab. We were making assumptions about some of the, the latest clinical treatment combinations and, some of the issues that we thought were relevant for the clinicians and the oncologists. And having him in our lab and at every lab meeting provided clarity on some of these issues, where he'd say, actually, we're not really that interested in that as a biomarker anymore, or the combinations now flip the other way around, or we put preference on this treatment. And then over the years, I've had more and more clinical fellows in the lab. And I think at the moment, I've got 6 clinical fellows, right through from young clinicians all the way through to senior consultants in the lab. They are the conduit between the work we do and the hospital and our clinical partners over there. So, we try to build it from within, but also we try to create as many networks as we can across the campus. And then going back to the social aspects, we have a social gathering in our institute at CRUK, and we call it Crisis. It originally came from CNRI, which was our original name, abbreviation. And we do this on a Friday afternoon. And what we decided, and it's not me, that's our director, Greg Hannon, a few years ago, decided that actually what would be far more effective is if we did the social event on the outside of the security barriers, where people can come and go. And when we could do that, we could open it up to the wider campus. And so what we've decided, and we've been doing this over the last couple of years, is that when one of the breast cancer labs in our institute is hosting crisis, because we do this on a rotational basis, we will invite all of our clinical colleagues over and they can come and mingle and we start chatting, and that's how collaboration's done about.
That's so exciting, isn't it, when you get all those different perspectives in one place?
Absolutely,
and it's so great because it keeps the research real and on track and impactful. And let's face it, that's what we're all trying to do here. So tell me a bit more about what molecular oncology is and why it's really important for the treatment of cancer.
Yeah, so molecular oncology is quite a broad term, but it basically means the study of the molecules in cancer. It's not specific to any one cancer, but molecular oncology is about trying to understand the proteins, how they interact with DNA, how they mediate cell cycle progression, so how they cause cells to divide, and how they contribute to cancer progression. And it really is about trying to understand the building blocks, how do the proteins fit together? How do they actually create molecular pathways that will cause genes to be regulated, switched on and switched off, and ultimately to cause cancer cells to do what they're going to do. A big part of that, from our experience, is developing methods, trying to understand how these really complex protein interactions form. And we know for some of these molecular complexes, there are up to 100 or 150 proteins that come together in this really beautiful coordinated process to build this complex. That complex is then an engine which can then switch on genes. How it switches on the genes and the genes that are going to be switched on and when is quite a complicated process. So trying to understand how these proteins, which ones are important, and where they go in the genome, and how they can regulate the genes. That's my interpretation of molecular oncology. And it really is a fundamental basis of not just breast cancer, but all cancers.
My next question was, is it relevant to all different types of cancer, and not just breast? Is it more relevant to some, or is it just relevant to all?
No, I think it's probably studied more in some cancers than in others. So we know in some cancers, we're very, very hyper-focused, and rightly so, on the mutations and the events that actually cause that cancer to become a cancer. And what we know, of course, as you know, and the audience knows, is that cancer is not one disease, it's 200 different diseases. There are similarities across some cancers, but there are cancers that have very distinct mutational signatures or perturbations that are very specific to that particular cancer or that particular organ. Now, we know that in some cancers that people spend sometimes their entire career studying signaling pathways, or how some of these mutations might be driving cancer progression. But ultimately, for a cancer cell to be able to divide, or to be able to secrete the signals it needs, or to be able to move and metastasize, to be able to colonize a secondary site, so to form a metastasis, it's got to be able to do function, right? It's got to be able to do things. So divide, move, colonize, communicate with adjacent cells. And a lot of that comes down to genes being switched on or off that tell the cell what to do. Those genes will make proteins, will degrade proteins, make enzymes, make structural things, secrete, make things that get secreted from the cell as signals. And these genes get switched on and switched off by these transcriptional complexes. And in my opinion, the molecular oncology really is studying those proteins and protein complexes, understanding how the Lego blocks fit together to form these big molecular structures that ultimately cause the genes to tell the cell what it's going to do. And that is fundamental to every cell type, not just cancer, but we see that every single cancer cell type. Now, what we know is that for many cancer types, we know less about the transcriptional complexes of the molecular events that occur. And in some cancers, like breast cancer, we know a lot more.
It is completely fundamental, isn't it? It is absolutely the building blocks.
Yeah, I agree.
So who do you collaborate with, and what challenges and opportunities does collaborating bring for you?
So we collaborate with pretty much everyone. So I learned when I was a junior scientist that if you want to study something as complex as cancer, you really need multiple people involved. You need to be having people that have different skill sets and different experience to look at the puzzle from different angles, and then working together in this harmonious approach where we share ideas and we can bat ideas back and forth. So we collaborate. I'm not even sure how many labs we collaborate with at the moment, but it's got to be active collaboration, dozens at the moment, and they are across the world. So we collaborate with labs, of course, in our institute and in Cambridge, around the UK. I collaborate quite closely with a number of labs in the US. I'm originally from Australia, so I've still got very strong connections there. We're collaborating with labs in Adelaide and Sydney, previously in Melbourne. And so we really collaborate with whoever we have to solve the puzzle that we're working on. And I think in the past, it used to be a little bit more difficult because of geographic distances and time zone differences. I think, as we all know, the world's changed dramatically over the last decade or so. And one thing I found, it's almost as easy to collaborate with clinicians in Australia or molecular biologists or chemists in Australia or America as it is to collaborate with somebody on the other side of the Admiral's campus. because the communication is much easier now. And so we really collaborate with the best people we can and the most appropriate people. And that really is determined by what the project is and what we're focused on and who we need to work with at that time. The one thing we have done is we've developed, as I mentioned, trying to study these protein complexes is a lot about trying to develop new methods to be able to study them. Like I said, these really complex protein interactions and the complex utilization of the genome to be able to switch genes on and switch genes off in a very coordinated fashion is incredibly complex. And that means you have to be able to develop methods to study the whole genome and to study protein complexes. And to me, really like doing method development. It's something that the reason I started in science, I really thought I was going to be a method developer, but just out of necessity, we realized we would hit a wall in our understanding, and to actually circumvent that and to be able to learn what was beyond or understand what was beyond the wall, we had to develop methods to be able to look over or under or around the wall. And so just by default, we ended up developing new methods to be able to study how proteins interact with the DNA, to mapping where they go in the genome. We had to develop methods to be able to identify what proteins are in the complex by using approaches where you purify one protein and use mass spectrometry to identify what its friends are. And then this turned out to be really, really powerful for us because when we would develop these new methods, it meant that we could look at the problem from a unique angle. So we could see the problem from an angle that no one has ever seen before. And almost always we would learn something new. And usually it wasn't what we expected or hypothesized. It was what we saw. And we really like doing that because we like to let the biology tell us what we should be looking at. And then we will develop the hypothesis based on what we identify. But one thing I realized is if you develop these tools, you become very popular because people around the world, if I go and present the data, and I like presenting data almost as soon as it's stable, well before it's published, I'm excited about the things that are going. I go to conferences, I want to talk about the most exciting thing. And a lot of that is around the methods that we've used to discover the new biological finding. And I've continued every time I've gone to conferences and people say, oh, there's a really cool method. I'd love to be able to collaborate with you. And so we collaborate with absolutely everyone we can. It gets to a point where we just don't have the bandwidth or the resources to be able to collaborate with everyone. So we try to enable other institutes and other labs. We will host people, train them in our methods. We will go to other institutes, train them. We will make sure all our methods are really carefully annotated and put out in the community. We have licensed methods to companies so that they can offer a service. And we try, of course, to do as many collaborations as we can. But in theory, we could just be working purely on collaborative projects and not doing our own work. So it's just trying to get that balance right. So we love collaborating with people. We work with as many people as we can, but it is just trying to get the balance right so that we don't undermine what we're trying to do at the same time.
It's really exciting to hear you say that, Jason, because so often I hear about the university that people work in silos and it's very difficult to connect. So it's really refreshing to hear this amazing sort of outward reaching and that, you share your methods and you train people and, rather than reinventing the wheel, you're just sharing your ideas all over the place. It's just brilliant.
I mean, I think cancer has been specifically, I think the old days of doing a really detailed biochemistry in a cell, just learning about fundamental biochemical processes, you could have one student or one postdoc and a supervisor. I think if you want to study something as complex as cancer, you need a team that's pretty complex. And I think it's very, very difficult to do it alone. Or you can't work properly, in my view. Not if you're trying to study the whole genome and these big protein complexes. You have to have experts in protein chemistry and computational biology and clinics. We've got chemists in our lab. And so I think you really, to tackle the problem properly and appropriately, you really need to have quite a multidisciplinary team.
Has your research been used in the clinic yet?
I mean, the obvious one is something we're going to touch on in probably a few minutes, which is the Pioneer trial. I mean, sometimes it's quite hard to make the link between fundamental discoveries and the impact of the clinic. But a long, long time ago, we made observations that the estrogen receptor in breast cancer can switch on a gene called cyclin D1. And we mapped how it worked. So we would bind to a particular part of the genome around the cyclin D1 gene. We would then bend the DNA to be able to contact the genome and switch cyclin D1 on. And at the time, we thought that was a fundamental biological discovery. We put it out in the literature. Then, of course, CDK4/6 inhibitors came along, and they work really well in estrogen receptor positive breast cancer patients. And it's because estrogen receptor switches on cyclin D1, and there's a hyperdependence on cyclin D1 because cyclin D1 is one of the downstream genes that allows estrogen receptor to do its job. And so the reason why the CDK4/6 inhibitors work is because it's blocking the cyclin part of the CDK4/6 complex. And so we didn't develop the compounds, but actually I think, some of our work made the fundamental link between estrogen receptor and the target gene to explain why CDK4/6 inhibitors work in ER-positive breast cancer patients. We have, we've contributed a lot to our understanding of the genes that are regulated by estrogen receptor. And a number of these genes have become part of these diagnostics, so these gene expression profiling diagnostics. these diagnostics, of course, are being developed anyway, but I think what we've done is we've provided some biological insight to why those genes are on that platform. And it's because they're targeting the estrogen receptor, either switched on or switched off. So we've provided some of the molecular, explanation. I think probably the more obvious example of how our work has made it into the clinic is the Pioneer trial, which has just finished. And, I'm really excited about that because that is the culmination of, 12, 13 years of work.
How was the Pioneer trial thought of? What was the basis of the work? And then if you could tell me about the trial itself and the outcome and the implications, that would be super.
Yeah, so I might actually break it up into different chunks because they're actually quite discrete parts of the story. So we developed a method called RIME, so RIME. We developed this probably about 12 years ago now. And it was a way where, as I alluded to try and study these protein complexes. Now, RIME is a method where you can use an antibody against a protein of interest, and you pull out that protein of interest, but then you use mass spectrometry to identify what it's associated with. And we had a little trick. It's a molecular glue where if a protein is part of a big complex, we can glue it to the Lego blocks together. We can pull out the Lego block that we're interested in and use mass spectrometry to identify the other Lego blocks that are in that complex.
Just tell me what mass spectrometry is, because my audience, my listeners might not know.
So mass spectrometry it's a way of looking at all proteins. So you run all proteins through the machine and you don't tell it what to look for. It will say, I found this protein and this protein and this protein and this protein. And there are tens of thousands of proteins. Each one of those proteins can come in dozens, sometimes hundreds of different variants that are slightly different. And what the mass spectrometer does is it tells you these proteins are the things that we've discovered. So basically, it's like going fishing. We throw the line in with a hook, we catch the fish that we want, but it turns out that fish is glued. We can glue it to its friends, and we can identify the particles and the other things that are stuck to that fish. And the mass spectrometry approach allows us to sift through everything, and it tells us, we've discovered this protein, we've discovered this protein. And we could suggest that these proteins work together, and then we could go validate that. Yeah. So it's a way of fishing for proteins. So we did this. So we developed this approach, and we used it to fish out estrogen receptor from breast cancer. Initially, cell lines growing in the lab. So these are just some models that we have in the lab, but ultimately we validated this from whole biopsy samples taken from patients who had breast cancer. And what we found was when we pulled that estrogen receptor, we found a really unusual protein that was stuck to it. And it was the progesterone receptor. Now, the progesterone receptor is a cousin of estrogen receptor. It's very similar. They've got similar structure. They have similar but quite distinct functions. And nobody really knew what progesterone receptor did in breast cancer. We knew it was a good biomarker. So if a woman had an estrogen receptor positive breast cancer and her tumor also expressed progesterone receptor, then she had a better outcome. And that's because estrogen receptor will switch on progesterone receptor. So it's one of the genes that's turned on by estrogen receptor. And so the theory is, and this is correct, is that if a woman's cancer has both estrogen receptor and progesterone receptor, then it means the estrogen receptor is really working because it's turning its target genes on. One of them is progesterone receptor, and it's on because the estrogen receptor is really working. And if estrogen receptor is really working, then drugs that block estrogen receptor are likely to be functional. They're likely to be effective. And that is correct. But when we pulled out the progesterone receptor as a protein partner of estrogen receptor, this suggested actually it might be a bit more complex than that. It might be that the progesterone receptor is doing something with the estrogen receptor. And then I remember the day my student made this observation, and he came and showed me, and I thought, that's interesting. But do we want to chase it? We had so many projects going on in the lab at the same time. But you know what? There's so much literature on progesterone receptor, but very little, almost nothing suggesting it did a functional role in breast cancer. So we thought, let's just do a couple of preliminary experiments. And those preliminary experiments ended up being about 4 years worth of work, is that, as is typically the case. And then we got to a conclusion where what we realized was the reason why progesterone receptor interacts with estrogen receptor is because it can influence what estrogen receptor does. And it turns out if you activate the progesterone receptor, now progesterone receptor is a protein, it's like a lock and a key so it is the lock, and it's only active when the key goes into that lock and turns it, and then it changes, it becomes active. Now, the key equivalent is progesterone, right? So progesterone goes into the progesterone receptor, it activates it, and then it becomes functional. Now, if we do an experiment where we treat breast cancer cells or patient samples with progesterone, what we found was the progesterone receptor, because it interacts with the estrogen receptor, it steals estrogen receptor and it hides it in the genome. It basically camouflages it. And we'd never seen this before, but and as soon as we got this piece of data, we realized, this is pretty interesting because we've got a new way of blocking estrogen receptor. Instead of trying to block it with a drug, we can hide it. We can bury it somewhere else in the genome where it's not doing anything bad, where it's not causing cells to grow, where it's not switching on this gene cycle in D1. And so we spent years and years validating that, showing that if we activate progesterone receptor, the cousin of estrogen receptor, we can indirectly block estrogen receptor by taking it and hiding it somewhere else in the genome where it's inert, it's not doing anything. So it's still there, hasn't been degraded. We haven't necessarily blocked it with a drug, at least in the lab experiment. But what we've done is we've inactivated it through this unique mechanism where we've camouflaged it. And we've hidden it. And so we spent a long time validating that, and then other people in the community validated it, and that really formed the basis of the Pioneer trial.
And is this important because then if you're not putting toxicity into a person, you're doing it, instead of treating with a drug, you're doing it in a slightly different way. So you're saving them toxicity and. So
yes and no. So I think in ER positive breast cancer, patients are always going to be taking the ER targeted drugs or their standard of care therapy. So this is in addition to the treatments that they're getting. But of course, as is the case for drugs that target estrogen receptor, because they are drugs that target a specific protein, we know how they work. They're targeted drugs. We understand how they work. We understand what the side effects are, and that only really affects cells that express that protein, unlike things like chemotherapy and radiotherapy, which will target every cell. So drugs that target estrogen receptor and progesterone receptor are only going to be doing things in cells that have the lock, because they're the key. So we did the preclinical work. We discovered that progesterone receptor could hide or camouflage estrogen receptor. We validated this many, many times in every single system we could. We teamed up with people around the world. They validated it as well. And then actually talking about our collaborations and some of the issues we had with our collaborations across the clinic at the beginning. So I gave a talk in San Antonio. It was a big breast cancer meeting called the San Antonio Breast Cancer Meetings in December every year. It's really big. There's about 10,000 people that go every year. And I gave a talk, it was in 2013, maybe 2012, 2013, where we just had a preliminary data on this progesterone receptor observation, and I presented it. And the feedback I got from the community after my talk was all very positive, and people were excited about it. And then one of my clinical colleagues, Richard Baird, who is really fantastic clinical trialist at Addenbrooke's, so he turns out he was in Texas as well. We both came back to the UK, and he contacted me and said, I was in the audience and I heard your talk in Texas, and we should work together. Well, that's not ideal that we both had to go to Texas.
Find one another.
Yeah, exactly. And I think that was one of the motivators, at least for me, to improve the relationship between the institute and the hospital. And we've definitely done that. But he said, look, I really see a clinical trial here because the drugs that are used to activate progesterone receptor, there's an old one, it's off patent, so it's cheap as chips, called megestrol acetate or Megace. It's been used as a cancer treatment, as a last line cancer treatment. Nobody really understood how it worked, but we do know in some women it works. And there's lots of old clinical trial data suggesting that it can be an effective cancer treatment. No one knew why. It was usually given right at the end because as a last resort, we really didn't understand how it worked. But there are drugs out there. We know the benefits and the disadvantages of these drugs. And why don't we set up a clinical trial where we combine an estrogen receptor targeted drug, so standard of care, but we combine it with this progesterone receptor targeted drug, this megestrol acetate or Megace. And the theory was, if the drugs that target estrogen receptor are 100% effective, fine. If the drugs that target the estrogen receptor, so these standard of care treatments, if they're not 100% effective, then by default, it's going to be some active estrogen receptor, and one of the genes it makes is progesterone receptor. So the very presence of progesterone receptor means that you haven't completely blocked the estrogen receptor. So our logic of the Pioneer trial was, actually, if we add a drug that targets progesterone receptor, it's almost like an insurance policy where if there's any active estrogen receptor in its cancer cell, we can camouflage it using this cheap off-patent treatment. And so we set up the trial. And I remember at the time, we were a little bit worried about whether we could get funding for this, because this was a trial that wasn't a licensed or on-patent treatment. This is it's not patent, it's an old treatment. And actually turns out that there are funders out there that will fund trials like this. We ended up getting funding from an organization called the Anti-Cancer Fund based in Belgium. We'll track my PhD student, Hisham Mohammed, who did all of the work on progesterone receptor, found and made the connection, got the conversation started. And then they ended up funding the clinical trial, the Pioneer clinical trial. And the trial been running for a number of years now. It was delayed, of course, as is the case for most trials because of COVID. But it finished recruiting at sometime last year, and it read out at the very end of last year, and the data looks fantastic. It looks really amazing. So we're still just processing the data at the moment. We are writing a manuscript, but the data looks fantastic. And if we include this megestrol acetate in combination with standard of care. We've done what's called a window of opportunity trial where women will come in, we'll take a biopsy, they'll be randomized to one of the treatment arms, and then a couple of weeks later, we'll take another sample, and we're using a surrogate of cancer growth. But then in that 2-week period, see what happens when we treat with either standard of care or with this progestin, what it's called progestin, this progesterone receptor drug. What happens in that 2-week window, and we can use an indicator of effectiveness based on that 2-week treatment arm. And it looks in the 2-week treatment arm that if we include the drug that targets progesterone receptor, we can improve the tumor inhibition. It looks fantastic, and across all of the arms. So we're really quite excited about this. We're still just going through the data at the moment and trying to understand the nuances of the data. But the trial's finished and it's positive, and we're just about to report it. So we're very excited about it.
I love that there was already a drug out there. So you're not reinventing the wheel. You just found something that was out there but wasn't really being used to the best effect. And you brought that in and you've used it to best. So it's just wonderful, isn't it? It's so kind of health economics wise, it's fantastic. Absolutely. For patients, it's fantastic. For the research, it's fantastic. It's just wonderful. Joining the dots, as it were.
Yeah.
So you've touched on it a little bit, but what impact do you think that the research is going to have on patient treatment outcomes?
Yeah, so this is the first trial. So I think it's going to require, a bigger clinical trial to make sure that this is to get confidence in this if we're going to include it in treatment arms. But I guess one of the interesting variables about the drug we use is megestrol acetate. It's already used to treat hot flushes. Now, women that are on aromatase inhibitors to treat their breast cancer. Aromatase inhibitors work because what they do is they block estrogen production from a woman, from the whole body, or a man. And depriving a woman or a man of estrogen has side effects in many, many tissues, because we know the estrogen receptor and estrogen is needed for bone mineral density and brain function and heart function. And so drugs that they use to block estrogen receptor to target the estrogen receptor in the breast cancer can be very effective. But by depleting estrogen from the whole body, you get side effects. And one of the main side effects of aromatase inhibitors is hot flushes. Now, these hot flushes can be really, really quite debilitating. And because women and men are on treatment for sometimes many, many years, that they're dealing with these hot flushes for long periods of time, and it affects their quality of life. Now, it turns out that one of the most effective treatments for hot flushes is the drug we use, but at a lower dose, at 40 milligrams. But we didn't know whether 40 milligrams had any anti-cancer activity, because the old literature suggested it could be used as a cancer treatment at 160 milligrams, but 40 milligrams can be used to minimize hot flushes. So our trial tested both concentrations. So it was standard of care, aromatase inhibitor plus the low dose of this megestrol acetate, the dose that's used to minimize hot flushes, and the dose that's used that we know has anti-cancer activity, 160 milligrams. Now, the higher dose of megestrol acetate we know has some risk. It's got weight gain problems. It has some risk of potential increased risk of blood clots. So we included the low dose as well, and it turns out that either dose looked like it had the same effect. So early days, but the low dose, the dose that's used for treating hot flushes, appear to be as effective as a high dose. So of course, the future work is going to be about trying to go and test the low dose and get to the hypothesis that we might be able to improve breast cancer outcomes through two different mechanisms. So one will improve the anti-tumor activity. So instead of just using an ER-targeted drug, we can use an ER-targeted drug, but also this insurance policy where any residual active estrogen receptor can be hidden in the genome, so we're camouflaged, but also to improve compliance. If we include this, so we can minimize some of these side effects, particularly specifically the hot flushes, and we improve compliance, and will women continue to take their treatment, one of the major side effects is being minimized. So yeah, so it is complex, but I think, so we're really excited about this, by the data. It is a pilot experiment. It's just a window trial, so we're going to have to do it along the trial, but aromatase said it, with all data is all pointing in the right direction. And so, we'll talk about doing a trial.
Will you go to the same funding body for that, or have you got more people interested?
I think it's probably going to be a bigger trial, so we might need to knock on some more doors. And I think I'm going to be relying quite heavily on Richard and his partners to be able to, and his colleagues to be able to, help make that part happen.
Yeah, amazing. One of the things I always ask people is, how is this treatment? Will it be used outside of Cambridge? How will it be used across the country, maybe globally? Do you have some thoughts on that?
Yeah, so this megestrol acetate has been used globally for years and years and years. And like I said, as a last line treatment, it is cheap. So I think in developing countries, clearly there's a space for that. It's not as Cambridge specific phenomenon. This is something that we think is going to be broadly applicable to breast cancer patients around the world. So yeah, not just Cambridge.
My last two questions. Obviously, I work with the Integrated Cancer Medicine Programme. So I would always ask, where do you think integrated cancer medicine will be taking us in the next 5 to 10 years? The idea of integrating all the data streams of a patient to give a more personalised treatment. What are your thoughts on that?
It might sound a little bit cliche, but it is actually genuine and honest. I think, we tend to put patients in really broad categories. So we know for some cancers, they have that cancer, and that's it. And there's no definition of different patients within that cancer type. I think in breast cancer, we've been much, much better at that. And I think in large part because we've been studying breast cancer for a long time. It's such a prevalent disease. It impacts so many families and so many people. And so because we've been studying it and there's been funding, we've been able to do molecular classification of breast cancer. And we now know that breast cancer is not one disease, but of these major subtypes, of course. I guess the fundamental ones are estrogen receptor positive, HER2 driven, and then triple negative. But even within each of them, you've got different subgroups. And I think at the moment, we feel quite confident that we can put patients into those major subgroups. But actually, we know even within estrogen receptor positive breast cancer patients, there are huge differences in treatment response. And we have so many treatments that block estrogen receptor, either directly or they will target something downstream of estrogen receptor, like these CDK4/6 inhibitors. And there's so many different combinations. We've also got new treatments that are targeting things upstream of estrogen receptor, things like PI3 kinase inhibitors, AKT inhibitors. And what we don't know is which treatments to use and in what order. So I think the integrated cancer approach, what it's going to tell us is by looking at a patient, we'll be able to get a fingerprint or a signature of that patient's cancer. And hopefully, what the ultimate goal is not necessarily to develop better treatments, but it's to be able to use the arsenal and use the treatments that we've got, but in the right order, at the right time, in the right patient. Now, at the moment, what so I think it's the things that we need to improve substantially is we can give women breast cancer treatments or men breast cancer treatments, and we don't know whether they're effective or not until, a period of time. And so maybe we see cancer come back. So I think one of the big issues is about detecting cancer, ideally non-invasive approaches. So we take a bit of blood, and there are huge numbers of strategies going on around the world to try and improve ways of detecting cancer early. But I think for the breast cancer community, it's about being able to identify, is that patient responding? And if not, what is the best order? And ideally, what we'd like to be able to do is to tailor the treatment and the order of treatment using the drugs we've got already, but tailor it to that treatment, knowing this is going to give the maximum impact in that patient, because we know that patient's tumor has this mutation and that copy number gain and this particular profile. And we know that with that specific profile, that this treatment regime and that order of compounds is likely to be most effective. So, in the breast cancer community, we have lots and lots of treatments, but it's just trying to figure out which ones to use, what combination and what order. And I think that this personalized breast cancer approach, I think, is really going to help us use the tools and the drugs that we've got in a much more effective way.
Yeah, that's exactly. It's so exciting, the idea that we can, use imaging and advances in all sorts of different fields in order to try and decide whether treatment is actually working, and then use that data for the next set of people that come along.
So I think it's a combination of things here. So being able to see the cancers and using imaging approaches, but also these cancer cells are spewing out things. So they're spewing out bits of DNA, fragments of DNA. They're spewing out bits of proteins that are specific to that cancer. And we have approaches to be able to look at those little fingerprints that come from the cancer, but being able to sift through them through piles of normal healthy cells or bits of DNA. That's the tricky part. But there are some really bright people around the world working on this. They're looking for these novel modalities and novel approaches to be able to detect cancer, in a quick, cost-effective and hopefully non-invasive way.
Yeah, absolutely. And so my last question really is, where do you see your research taking you in the next 5 to 10 years?
Yeah, that's a good question. I think for us, actually, we're almost taking a little bit of a step back. So one thing I haven't spoken about today is one of the discoveries we made when I was a postdoc in the US. I spent 5 years over in America, and I made a discovery there with my supervisor, Miles Brown. And what we discovered was that the estrogen receptor pathway in breast cancer is reliant on this very fundamental foundation block. And it's a protein called FOXA1. And FOXA1 is the very first piece of Lego in this whole pathway. And then the estrogen receptor is totally 100% dependent on FOXA1. Now, one thing we know is that a lot of the treatment-resistant mechanisms that we're seeing, things that we've seen for years and new ones that are emerging at the moment as better treatments come along, almost always mutations or commonly mutations or changes in things that make estrogen receptor become active when it shouldn't. So they are things like mutations in estrogen receptor itself, changes in downstream genes, changes in things like that hyperactivate estrogen receptor, so it's on more than it should be, even if there's a drug there. But what all the lab-based data tells us is that this protein FOXA1 is still there, and it's still absolutely essential. So, the way we see it is if we can block this protein FOXA1, instead of breaking the branches off the tree, we can cut the tree down at the base of the trunk. That's the theory. That's what the biology is telling us. Now, the problem is FOXA1 is a technical nightmare. It is very difficult to work on. It's difficult to study. It's difficult to look at. And I think that's just because of the nature of what it does. It's a very, very unusual protein. But we've been looking at this for 20 years. In fact, I think the discovering FOXA1 and linking it to breast cancer was why I got my job. I mean, it was that discovery that got me the interview and then ultimately the job. And we've been really interested in trying to study this protein FOXA1 for about 20 years now, maybe a little less, almost 20 years. And one thing we've done over the last year or so is what we've done is we've taken a big step back. And so we want to go back and dig into the fundamentals of this protein FOXA1, really try and understand every single nuance, every little part of the protein. What does it look like? How does it sit on the DNA? How does it recruit estrogen receptor? Are there vulnerabilities and little kinks in the system that we can use to our advantage? But of course, we can't target this unless we know what it looks like and unless we know how it functions. And it's very much, I use the analogy of the car engine. If the car breaks down, you're not going to repair it unless you know how all the cogs fit together and where the floor is or the brake pad is. And if you go replace that part and the engine starts again, we're in a very similar scenario where we have this protein. We know it's absolutely critical. We know very little about it. So what we're doing is our lab is studying, we do some really fundamental work around this one protein. So really hardcore, molecular characterization and molecular oncology. And I'm enjoying it. I haven't done that in a long time. So really detailed. We have protein work and how does it fit on the DNA. So we're doing some pretty fundamental stuff. And that's probably going to keep us going for the next 5-ish years with the assumption that if we're successful, then we might identify a way of blocking this foundational protein, which, we think if we can do that, I think it'll be a real game changer. And there are lots of people around the world that have come to the same conclusion based on the work that the community have discovered, and we've contributed to that. So that's going to be one big part of the lab. We're also starting to do work in other cancer types. So we've always had an interest in prostate cancer because the parallels between estrogen receptor positive breast cancer and prostate cancer are really substantial. So prostate cancer is driven by a protein. It's not estrogen receptor, it's androgen receptor. The key that goes into the lock is not estrogen, it's androgen, but it actually uses very similar mechanisms to sit on the DNA, to switch genes on and cause prostate cancer cells to grow. It also uses FOXA1. So it turns out FOXA1 is used in both breast, prostate, and it turns out that pancreatic cancer will switch on FOXA1 and will use it to drive metastasis. So this protein we've been studying in one context, we've always known it's also doing something in prostate cancer, so we're really digging into that. But some recent data that's come out of the US suggests that pancreatic cancer have figured out a way where they will switch it on when it's normally not on, and then it will become a contributor to metastasis in pancreatic cancer. So we're definitely heading in that direction. And we spent a lot of time building toolboxes and ways and methods to be able to study these difficult proteins. And we are going to, of course, apply it to breast cancer, estrogen receptor positive breast cancer. But we are also really interested in leveraging the insight, the experience, and the toolbox we have to look at this protein in some of these other contexts, not just studying breast cancer and the fundamentals, but it's about looking in prostate cancer. And more recently, we've been doing some work in pancreatic cancer. That's keeping us busy. And all the projects are very different. They're pointing in different directions and really telling us very distinct things. And we're having to chase those things depending on where the biology tells us we need to go. But it's a challenge and it's exciting. And every day we're doing something new.
So really back to basics for you then for the next 5 to 10 years.
That's right.
Brilliant. Oh, Jason, it's been so interesting to talk to you. Thank you so much for taking time out of your day.
No, thank you very much.
If you want to find out more about the work of the Mark Foundation Institute for Integrated Cancer Medicine, please visit our website at www.integratedcancermedicine.org, where you can find details of the ICM vision, all the current research, clinical trials, resources, publications, and team information. You can keep up to date with our latest news and events, and you can also sign up for our newsletter. If you would like more information about the work of the CRUK Cambridge Centre, please go to www.crukcambridgecentre.org.uk, or you can connect with us on Twitter using our handle @CRUKCamCentre. Thanks for listening, and do join us again soon.